lesson, absorption by roots, the processes involved. This is again a lesson for standard 10 biology. Now let's see, I have mentioned here the word tonicity. And here you can see the thistle funnel experiment is set up here. Now let's say, we say inside the thistle funnel we have 5% sucrose solution. And outside we have put water. Now what would happen after a while? The water molecules through this semi-permeable membrane. Let us say this semi-permeable membrane here is parchment paper. It acts as a semi-permeable membrane. Hence the water molecules from their region of high concentration will move in through the semi-permeable membrane and they will come into this solution. So if the initial level was here, after a while the final level will be here. Now if for some reason we think that all water is moving inside, but what we can do now, we can reverse this also. We can put 5% sucrose solution outside and fill the thistle funnel with water. At that time what will happen? The water molecules from inside the thistle funnel will come out into the beaker. Here, this will be sucrose solution and from inside the water molecules will come out. So what happens? That brings us to tonicity. What is tonicity? It's defined here. Relative concentration of the solutions that determine the extent and direction of diffusion. Now see what is happening here. Outside, we have changed the concentrations. First we tried by putting the sucrose solution inside. Then and water outside. Then we put water inside and sucrose outside. So every time water molecules, because it's a solvent, its molecules moved from their region of high concentration to their region of low concentration. So wherever there is this concentration, relative concentration, what would happen? That determines in which direction the water molecules or the solvent molecules will move. Now that brings us to some more important terms. Let's say, I say isotonic solution. Solutions on both sides, or you can just say isotonic. Now what happens here? Suppose I say here also I am keeping water inside here, water on both sides. Let's try different combinations. Water inside, water outside. Will there be any osmosis? Will there be any movement of water molecules? No, because the concentration is the same on both sides. So there will be no movement. So we can call this as isotonic. This is isotonic. Remember this word isotonic very well. So concentration, relative concentration on both sides is the same. Inside the parchment, one side, it's separated by a parchment. On one side of the parchment, on the other side of the parchment, the concentration is the same. So hence, end result will be, there will be no osmosis. No osmosis, no movement of water molecules in an isotonic. Now suppose I say hypotonic. Hypo means lower. So what is happening? In the beaker, outside the thistle funnel, we are putting water. And inside, we are not putting water. This time, we are just putting a sugar solution. Let us say 5% sugar solution. In this condition, what will happen? 5% sugar solution. What is happening? The water is moving in towards the direction from its direction of high concentration towards direction of low concentration. So there's inward movement. Hence there is endoosmosis. Going inward. Endo means going inward. Then we have another condition where we'll reverse this. We will put it as, we'll use water inside, water here, and 5% sucrose solution on the outside. That condition, what would that condition be called? Hypertonic. Hypertonic. What is happening here? So what will happen? The water will move out. Hence there will be exosmosis. Exo means outside. exosmosis is happening. So understand that this is called tonicity, relative concentrations. And what do these concentrations do? They determine the extent and direction in which diffusion is going to happen. Now let us see these. We can divide the solutions into these three types: isotonic, hypertonic, hypertonic, hypotonic. I had mentioned to you here, no osmosis is happening here, endosmosis and exosmosis here. Now let us see why this is happening. Suppose we are saying the relative concentration of the solute molecules on either side of the cell membrane is same. Now let us take a container and let's imagine that we have placed a cell here. And it is in some kind of a solution where the concentration of the solute on either side is the same. What will happen? No osmosis will happen, because both have the same kind of solute molecules, the same concentration of solute molecules. Here the cell membrane here is acting as a semi-permeable membrane. So even if we say that two solutions that are separated by a semi-permeable membrane, and if they have the same concentration of solute molecules here and here, then it can be used in the term of the solvent molecules also. Then we say the solution here, both the solutions here are isotonic. It's a relative thing, it's a comparative thing. Hence, no osmosis happens. Now if we, then let's come to the next one, hypo. Hypo means low. Relative concentration of the solute molecules in the solution outside the cell is lower. Solution outside the cell is lower. So what will happen? Of what? Of the solvent outside the cell is lower than the fluids inside the cell. Then what will happen? There will be endosmosis. There will be endosmosis. That means it will move in. Endosmosis. Then hypertonic solution. We are talking about a solution where both the solutions comparatively, where the relative concentration of the solute molecules in the solution outside the cell is higher. Now this time outside the cell is high. So in this condition, what will happen to the cell? It will move out. The solute here we are talking about the solute moving out. So these are three solutions which we can categorize them like this. We can compare them on the basis of what processes are happening. Osmosis is not happening here, nothing is happening here. Endosmosis is happening in this condition, exosmosis is happening in this condition, and their relative concentrations were on either side of the semi-permeable membrane. Now here we spoke about a cell, so the cell membrane here is the semi-permeable membrane. Now let us look, here there are some simple diagrams, please take a good look at them. Now we have placed here an animal cell in an isotonic solution, in an hypotonic and hypertonic, and here there is a plant cell being put through the same solutions. Now look at the animal cell. What happens if it is placed in an isotonic solution? What would it mean? That the concentration on both sides is the same. So the amount of water entering in is same as the amount coming out. So hence the cell shape, the cell size remains the same. Now if the cell was placed in a hypotonic solution, that means what? The concentration outside is low. So what would happen of the solute in comparison to the inside of the cell? There is exosmosis. See how it is leaving, and amount entering in is less. Hence exosmosis happens here. Exo will happen here. Exosmosis. See the condition of the cell. It is shriveled up. It has become it's not flaccid and it's not bulging, it's not round, it is shriveled up. In an hypertonic solution, what would happen? The water entering in is more and leaving is less. So what is happening? See, the cell has ruptured. It has put so much pressure on the cell that it has ruptured. So you can see, this is actually an RBC which we have drawn here. You can see the fate of the RBC cell when it is placed in different kinds of solution. Now look at the plant cell, what is happening. In an isotonic condition, see the isotonic solution, the vacuole is so large, it is full of vacuolar sap. Entry and exit, amount entering, amount leaving is the same. Hence the vacuole is very full of sap, and it is in a balanced state. Here what is happening? In a hypotonic solution, the water leaving here is more than the one entering. So see what has happened. The cytoplasm has left the walls here. This is the cell wall. The cytoplasm has left it, and the cell contents have all come together. They have shrunk together. Look at the condition of the vacuole here. See, it is also small and little distorted like this. So they have the side. All these contents have left the cell wall here, and they are come inside. This is the plasma membrane. So that is how what happens to the plant cell when it is placed in a hypotonic. Now you can see very clearly it has a cell wall. So overall it hasn't become like this because of the cell wall which is rigid, and it's allowing it to keep its shape. Now here see, in a hypotonic solution, look at the vacuole, full of vacuolar sap, and the walls are bulging. Rupturing can be less here. Why? Because the rigidity of the cell wall is allowing it to maintain its shape to some extent. So entering is more here of water and exiting is less. So it is fully full. It is full in a turgid state, we can say, and it is bulging. But the cell wall is helping it to retain its shape. Look at the vacuole again. See how full of sap it is. Let us now study active transport. Now I have drawn for you a simple diagram showing the root hair. Now what happens here, inside the root cells here, the concentration of sulfates, nitrates, potassium, manganese, zinc, all this is high, and it is higher than the conditions outside the root. Now why this is higher? Otherwise there would have been no osmosis happening, water would have not entered. So the cell membrane is here, it's keeping it inside. The concentration is high, and it's for a reason. Why? Because we need to have osmosis happening here. Outside it's low, but still these are nutrients. They are required for various functions of the plant cells. They have to be taken into the plant cell. Now for that reason, nature has made an arrangement here that these still enter in. How they go into the cell? How? Because it's required by the plant. So various processes happening inside the body of the plant. Now, it is taken in forcibly, and what is being used? ATP, energy is being used here. So whenever a process requires the use of energy, ATP, that time we say it is active transport. Here these are being forced to enter into the cell, though the concentration inside is higher than the surroundings here. Now then that brings us to another word, passive. Passive transport. What would that mean? Opposite of active transport. Here ATP is not being used. ATP not being used. So it is just happening, not being used. So to understand the difference between passive transport and active transport. So you can be asked to compare the two. Let us study this next phenomena, turgidity and flaccidity. Now here is a simple diagram of a plant cell. You can clearly see the structures of the plant cell, the cell wall, the plasma membrane. Now can you see different kinds of arrows that have been drawn here? What do they signify? Here I have mentioned osmotic pressure. It means water is entering in. Water is entering in. Why? Because of the concentration gradient, inward. The water is moving inward, entering. When water enters in, the cell here becomes fully turgid. That means it is in no more water molecules, and we can say there will come a stage where no more water molecules can enter. That means the cell is fully distended, it is fully stretched, the wall is completely stretched, and it is water, any more entry of water is stopped, even though the concentration inside is still higher, so they cannot be, it is fully stretched. Now, and in that condition, here the contents of the cell, they will they'll put a pressure on the wall. Because it is full of water, it will put a pressure on all directions. So this pressure is called turgor pressure. See, it is acting towards the outer side, it is pushing the wall because it's full of water molecules, or hence it is an outward leader. Look at the vacuole, fully stretched in, completely filled with vacuolar sap. Then we have the wall pressure. At the same time, the cell wall, because it is the turgor pressure is acting outward, the wall also pushes the cell contents with a certain pressure. That is called wall pressure. So let us define this turgor pressure. When a cell is in a fully turgid condition, the turgor pressure, it acts in an outward direction, wherein the cell contents apply a pressure, put a pressure on the cell wall. At the same time, what is wall pressure? Wall pressure is the pressure exerted by the cell wall on the cell contents. See, you can see it is working inwards, this is working outwards. So, understand the difference between turgor pressure and wall pressure. Also, see in this diagram, if you are given a diagram like this, you should be able to identify the direction in which both are working. Look at this diagram of a plant cell. So, you can see this is the cell wall. Cell wall. And here is the plasma membrane. Plasma membrane. Now this cell, just now I told you about turgidity, it is in a fully turgid conditions. But if this cell is placed in a solution that is hypertonic, that means what will happen? This after a while, now it's in the beginning, it's in a turgid state. After a while, this plasma membrane will separate itself from there will be shrinkage. There will be shrinkage because water, fluids from inside here, from the vacuole and from the cell will move outside. If it is placed in a solution whose concentration is higher than the concentration of this cell, of the fluids of the cell. So what will happen? It will leave the walls like this. So if you are told to draw next diagram, you might as well say, see the plasma membrane is now leaving the walls of the cell. This would be your next diagram. So this is the plasma membrane. Everything is shrinking. The vacuole even may become a little more smaller. Okay, and finally smaller and smaller till it will reach a stage where you will see that this interior, these contents along with the plasma membrane have completely left the walls here. They see the last, in the end, like this. This is the cell wall. So these contents have completely shrunk and come to a state, the vacuole is hardly there, and it has completely separated itself from the, so in this condition, see these are the inside the contents with the plasma membrane, it has completely left the wall. So it is said to be in a flaccid state. It will become soft and shrunk, and it will be in a flaccid state. Now see, suppose we have a football here. Suppose I am drawing a football here. Now suppose I say that the outer wall of the football is the cell wall. Imagine in your mind that this is the cell wall and the plasma membrane is that rubber bladder which it has inside. So when the rubber bladder is full, it is completely sticking to the outside the skin of the football. But if this rubber bladder gets deflated, so it gets deflated means it lies inside like this, and it is totally separated from the cell wall. In this case, you can think in your mind as if it is a football. So this would be the plasma membrane if it was a cell. Membrane. That brings us to this condition called flaccidity, where all the cellular contents have shrunk and they have come towards, the plasma membrane has left the cell wall like this. Now, if this cell, which is completely plasmolized, means everything has shrunk and it has become flaccid. This cell has been plasmolized. If it is put back in a solution that is hypotonic in nature, that means it will again come back and it will deplasmolize. It will change itself and it will come back to its original shape. That means all the cell contents, the vacuole will come back to its shape and it will completely fill in the interiors of the cell. How turgidity is useful to plants. So it provides rigidity to soft tissues like leaves. You can see when a potted plant is kept outside in the sun because of excessive transpiration, loss of water from the surface of the leaf, what happens in the end? They become very droopy and flaccid. Now again in the evening when the plants are watered, they become rigid again because the water enters into them and it brings them back to their shape and helps them with their various processes also. Now, roots can push through the soil. If the roots were soft, they would have not been able to, if they were flaccid, the root cells, it would have not been able to grow into the soil. It builds up root pressure. Now, as it builds up root pressure, it helps in the water to rise up. We'll do root pressure after this in detail. So, the water rises up to a certain extent. It helps to carry the water in the upward direction. Opening and closing of stomata. You know, the stomata is protected by guard cells. So, these guard cells, they control the amount of gases being diffused and, helps in various processes of the plant. In photosynthesis, they play a role. In respiration, they play a role. So, opening and closing of these guard cells is also because of this turgidity. So, they become turgid, they open, and when they become flaccid, they close again. Turgor movements. Now, you know plants like, let's say, touch-me-not, mimosa, pudica, these kind of plants. These plants, when we touch them, they are sensitive to touch, they do. But then how they come back? When the cells become turgid, they come back after a while to their origin. So, turgidity and flaccidity, it controls these kind of movements. Now, there is a term here called bleeding associated with root pressure. So, if you cut a stem, you will find fluids coming out of it. That coming out of fluids, it's called bleeding in plants. Let's study root pressure now. Here is a simple diagram. This diagram doesn't have many details so that it becomes easy to understand. You can see here, this is the section of the root. So, if this is the root, it has been cut here. So, you can see this is the root hair. Root hair surrounded by soil particles, and in the soil particles there are these water droplets here. So, it enters, the water droplet enters the root hair, and this is the path it takes. Now, as it is going, this is the xylem here. You can say xylem. Why we are mentioning the word xylem here? Because xylem is mainly responsible for transportation of water. Now, what happens here? This part is the endodermis. Somewhat like this. So, as the water travels from the root hair across the thickness of the root, you can see from one cell to the other, the water is moving till it comes here to the xylem, and then it goes up. Now, you can see there will be a simultaneous expansion. As the water enters, this cell will expand, and then after the water leaves this cell will expand, and this cell will be little, it will become little flaccid. So, water is entering, it is becoming turgid, and when the water is leaving, it is becoming flaccid. But this happens fast. So, there is a simultaneous wave of expansion and contraction of the cells. This generates a kind of pressure. This pressure is root pressure. Suppose, and this is very good if this pressure works well in small herbaceous plants. It actually helps, it generates a pressure wherein the water rises. Now, let's see if this wave of expansion of contraction was not there, the water would have not been able to move from one cell to the other. So, one becomes turgid, the other becomes flaccid, one becomes turgid, and then so on and so forth via the xylem. Now, if we take suppose we say take a herbaceous plant here, and this is the plant growing, and the plant has been cut, this can be, and you put a pipe here with a graduated scale, you can actually measure the root pressure. You can actually measure the root pressure. This pressure actually helps in transportation of water along the thickness and upwards into the plant. So, it is one of the major factors which allows movement of water or inward movement of water, and then thereby pushing it up through the stem. Now, you see sometimes in the mornings when you look at your plants like money plants and other plants, you will find here there is a leaf. On the tip of the leaves here, there will be droplets of water. Now, here see these droplets, this tip of the leaf, this part is called the hydatoid. Now, here there will be a droplet of water like this, see, here on the tip of the hydatoid. This process is called guttation. So, loss of water, or actually this is not water, it's a sap. So, loss of this sap comes out here and rests here, especially on cooler mornings, you can see. through the hydatodes. That means on the tip of the veins of the leaf, these hydatodes are there. Through them, this sap comes out. This process is called guttation. Now look at this. This is the structure again of a root hair with the adjoining cells this way. These are the xylem. Xylem. This is the root hair. These are plant cells, hence they have their cell walls, large centrally placed vacuoles. Now are surrounded by soil particles with water between them. Now what happens? Let's sum it up. From the soil the water enters because there is a difference in concentration. So the water enters into the root hair by the process of osmosis. This starts a continuous wave of expansion and contraction. The cells become turgid. When they are full of water, this is and then the water is passed on to the next cell because it's flaccid. So a concentration gradient is set up. So it helps in inward movement of the water till it reaches the xylem. Also, this minerals are pushed in by using ATP. Because minerals are required by the plant. Ideally, they should have not entered because there's a concentration is high inside, but they are pushed in using ATP. So active transport. So this is how the root hair, it carries out not only helping in absorbing water, also in taking in the minerals which are vital for the processes that are happening in the plant. So you can, as you see the diagram, you will understand how important the role of the root hair and the root and the cells here is to the existence of the plant. Let us now study and sum it up, the forces that contribute to the ascent. What is the meaning of the word ascent? Rising up, going up, of sap. Let's see, root pressure. So what happens? There is simultaneous contraction and expansion of cells from the root hair right into the stem, and then thereby causing the water or the sap to rise there upwards. In the xylem vessel, the water rises up to a certain extent. In herbaceous plants, this root pressure plays a major role in rise of water up. Then there is capillarity. So the narrower the vessel is, the higher the water will rise. This is called capillarity. Then we have transpiration pull. Now, as a plant, suppose there's a plant here, there's leaves on top. So it forms a continuous stream of water going up. As the leaves lose water this side, via transpiration. At the same time, because the water molecules are adhered to each other. They are sticking to each other. They are sticking here to each other. So as from here there is a loss, from here there will be an entry of water, adhesion of water molecules. So these are the major factors that contribute to the ascent of sap. The root pressure, the capillarity, so that along the walls it goes up of the xylem because they are narrow vessels. So the narrower the vessels, the higher the water will rise. Then the transpiration pull, which is caused by loss here and water entering in through because of the forces of adhesion. The water molecules show adhesion, the molecules stick to each other and they go upwards, forming a continuous stream of water going up.